Okay, good afternoon everyone. Hopefully all of you can find your seats if you're over in the poster session or in the back. We're just going to start our final session for the day where we're going to be having a variety of canola research updates. So my name is Chris Mancher. I'm with the Canola Council of Canada, and I'm going to be your chair for this session, as well as being the chair for this year's poster program, as well as our Flash Talks program. Last year, we introduced this session for Canola Week for the first time, and building on this success for this poster session, we're going to introduce it again this year, where we have about 2/3 of our presenters being students, and the other non-students and some other companies as well that have startup pitches on the last day of Canola Week. So, with our poster subcommittee for this poster session, we were able to select a handful of these students to present Flash Talks for this. So we have 7 lined up for us. And here we have, they're all going to be in front here for it. Li-Ping Wang with gene editing of starch metabolism enhances canola seed yield in both greenhouse and field conditions. I'm going to list off all of them right now, and then what we're going to do is they're just going to come up one after another, kind of like a blitz round for this, so that way we don't have to keep swapping places. So, Mohammed Mukhtar, an applied approach to verticillium stripe management in Manitoba, a proof of concept study. Muhib Ullah, high resolution ecological monitoring of flea beetles in canola using smart insect traps. Suman Parjuli, phenotypic and histopathological evaluation of the canola lines carrying clubroot resistance of Brassica oleracea. And Virginia Miguel, from Arabidopsis to canola, CPK28 homologs in pathogen resistance. And finally, Jihong Du, understanding of lipid metabolism during cold germination and seedling establishment of canola. And just as a heads up for our Flash Talk presenters, we have about 5 to 7 minutes to present your 3 to 5 slides. And I'll give just a nice little wave to you saying, okay, time to start wrapping things up, get about a minute left, and then we'll roll into the next presenter. And then after that, I'll come back on to announce our research updates from our provincial grower commissions. Okay, Li-Ping, all ready?
Hi, good afternoon, everyone. Thank you for the organizing committee for giving this opportunity to this talk. I will be giving a brief overview of our canola project. So as you all know, in oil seeds, the starch is not stored in the mature seed, but starch is very important for the plant development. Because about 30% of the photosynthesis-derived carbon is formed in the form of a starch. This starch are transitory starch, so it undergoes diurnal turnover and provides a steady carbon supply for the plant growth. During starch synthesis, starch branching enzyme is a key class of enzyme required for starch synthesis. So in the Arabidopsis wild type, as you can see here, so in the Arabidopsis wild type leaf tissue, the starch is darkly stained. But in the absence of SBE enzyme, there is no starch at all. In maize, which is a monocot, there is another different class of enzyme, which is specifically expressed in endosperm. So our initial hypothesis was simply to test whether these endosperm-expressed genes could function in Arabidopsis leaves. That's how we transferred these maize genes into Arabidopsis SBE mutant. So surprisingly, the results showed that those endosperm-specific genes could complement the starch synthesis, but also it led to large plant size and also markedly increased in seed yield. So this finding was really a serendipity for us. Due to highly conserved gene function between Arabidopsis and canola, we transferred this technology to canola. There are 2 steps. So the first step is to generate canola SB mutant using CRISPR-Cas9 system. We have obtained a range of mutations with different combinations. So 4X is quadruple mutant, which carries mutations in 4 genes, and the 6S carries mutations in 6 genes. I have to mention that there are a total of 6 SB genes in canola. Our 6X, there is 1 mutation still has a frame shift causing residue enzyme activities, so this 6 knockout still has residue starch content. So we are using 4X and 6X as genetic background for the second step to overexpress maize genes in this materials to generate transgenic canola. Now this is an image illustrating the transgenic plants overexpressing maize genes in the greenhouse. So you can see transgenic plants are much more vigorous than wild type. These values are an average of 10 plants grow in greenhouse conditions. So you can see the seed yield increased by 35%, and we also observed an increased number of branches of silic number by 40%. There is also a notable trait, which is a thick stem. So this trait has been observed in the 6 sectable mutant, and this trait also kept in this transgenic mutant. So it might confer a potential advantages for drought and heat tolerance. We conducted a field trial this year with support and assistance of Corteva Agriscience in Rockwood, Ontario. The yellow box showed the size of 1 plot. We have 3 field replicates, and every plot is planted with 1,200 seeds. So the 4 images is 4 stage. The picture was taken in the field. So the field trial data is promising. We have a total of 12 lines, including 2 controls. So the first control is DH1275, which is a wild type. The second control is a negative segregant. So this control has exactly the same genetic background as DH1275, but it underwent tissue culture procedure. This might affect the plant performance in the field, which is true in our field data. As you can see in this graph, the green bar indicates the negative segregant. So it is slightly lower than the wild type DH1275. So when we compared all our transgenic plants and gene-edited lines with this negative segregant, you can see most of the transgenic lines and the gene-edited lines expressed statistically increased seed yield. If you look at the number, it's 14 versus up to 21 bushels per acre. So this is 50% increase. Even though when compared with the wild type DH1275, most of the lines are increased, has increased seed yield compared to this line. So some of them still has statistically significant high. So this is still 25%. The seed quality and the oil quality, protein amount, there's no significant change. We have presented this in the poster, so you can visit the poster number 15. We would like to acknowledge the funding support from Canola Council, and also support and assistance for the field trial with the Cultivar AgriScience. Yeah, thank you for your attention.
Is it changing? Is the slide is showing there? Okay. Good afternoon, everyone. I am Mustafa, a research technician working with Dr. Harmit Singh Chawla at the University of Manitoba. With verticillium stripe continuing to spread across Manitoba canola fields, we carried out a province-wide assessment to gather field-level insights. So I am here today at Canola Week to share what we have learned from that work. In this talk, I'll start with why this work was needed, then what we exactly did on the farm, and also what we actually found across the sites when testing for verticillium, and then finally wrap up with key takeaways. So let's begin with why this study matters. Verticillium stripe was first detected in Manitoba in 2014, and now it shows up to more than 60% of canola fields. So it causes stem striping and plant weakening, and it can also lead to yield losses. As there are no registered fungicides and also limited resistance, we needed a better picture of what's happening in real field conditions. So our main objective is to understand verticillium levels under field condition across the province and see whether harvest method, season, or any other management practices were influencing the pathogen load. Let's look at what we did first. The Manitoba Canola Growers Association collected the soil and stubble samples from farms across Manitoba. And those samples, we actually dried them and then processed them and tested each sample using qPCR, which is simply a DNA test to test the, to measure how much verticillium is present in each sample. So once we had those numbers, we compared the verticillium levels across season and management practices to see actually what those patterns showed up. Once those samples were processed and tested, here's what we actually found. On the soil side, which is shown on the left side, we were able to see verticillium in all the 100% of the samples that been tested. Following that, fall soil samples had significantly higher verticillium levels when compared to spring samples. So with that, which fits with the pathogen building up throughout the growing season. On the right side, we looked at harvest method and yearly season fungicide application in the stubble samples. So neither straight cut versus swath in the harvest management side, nor the treated versus untreated in yearly season fungicide application made a noticeable difference in verticillium levels. So what really stood was the larger site-to-site variation. So that tells us that local field conditions and history likely have a bigger influence than any single management practices in a given year. Here we are looking at how verticillium levels at harvest lined up with yield. Across farm, whether they were part of the harvest management trials or the early season fungicide trials, we saw the general pattern. As verticillium load increased, we were able to see the yield actually decrease, but those relationship wasn't statistically wrong. Wasn't statistically strong. So visually we can see a downward pattern, but it isn't really reliable enough to predict the yield loss itself. The final takeaway from this slide is that verticillium can contribute to lower yield, but field-to-field factors have much higher impact than the influence of the pathogen alone. So to wrap up, verticillium was found in all the samples we tested, but the severity of those were varying site to site. We didn't see any single harvest method or early season fungicide application were lowering the pathogen or improving the yield. So this tells us that verticillium influenced more by overall history and also the local field conditions. So finally, we need a multi-year work which will help to strengthen the analysis and also to predict a trend. So I would like to finally thank Manitoba Canola Grower Association for their support in this research, and thank you everyone for your time today.
Good afternoon, everybody. My name is Mohibullah, and today I'm going to present high-resolution ecological monitoring of flea beetle in the canola fields using our designed smart insect traps, which we call SIT. As we know, insect ecology is actually understanding and investigation of the emergence pattern of the insects when they become active in the field and what time, and how the environmental drivers influence their activity and their behavior. It also analyzes the spatial distribution and movement of these insects across the field, and finally, how the pressure of the insect changes when the crop develops. So to capture the complete ecological picture, we actually designed the following strategies. We placed a network of 8 SITs, the smart insect traps, in the canola fields, in the AFC field here in the Saskatoon, to collect high-frequency temporal data. The aim was to monitor and understand when and where they become active in the field. Then the seasonal coverage strategy is started from early May, and we started monitoring, continued monitoring until the mid of September. So the spring emergence actually captured the initial emergence of these insects from the overwintering sites, and then the growing season tracked the population activity of these insects and how they interact with the developing crop. Finally, the fall transition, it captured the overwintering actually behavior, how they decline, and what was their relation with when the crop, the pod actually matured. So we also implemented this adaptive camera positioning to not only capture temporal data, but also capture the vertical movement of these insects. Like during the early season, we set up the camera near the soil surface to catch and capture those early season activity, and then later on set it up as the crop developed. So these are some of the key findings of our research. Regarding the temporal patterns, Flea beetle consistently emerged in the mid-May, which is labeled as week 2 in this heat map. And then they continued. These were triggered by specific humidity and temperature threshold, which I'll talk about in the next slide. The peak activity occurred from week 13 to 17, and that occurred within the canola plots. We actually placed these SITs in different locations to capture like their how they disperse in the field, from where they emerge and where they go. So, and then the activity declined after week 18. That became the the end of August. That was the time when the pod matured. The spatial analysis also revealed significant distribution dynamics. The activity was very high in the early stage near the grassy area, and then some locations like where the plants, the crop was near the grassy edges. We actually consistently showed consistently high activity compared to other regions like near the ones near the trees. There was a clear dispersal pattern found in their activity from the grassy area to the canola plots. Similarly, the environmental responses identified critical thresholds. We actually saw that with high humidity, even when the temperature was as low as 5, there was high flea beetle activity. And when the humidity was lower, the activity and the temperature was around 5 or less than 5, the activity became minimal at that time. The peak activity event come when the humidity was greater than 70% and the temperature was around 19. So during that time, we captured 350 flea beetles within 40 minutes of time. So by identifying these critical environmental thresholds and spatial temporal patterns of these insect activities, this research actually aims to provide a data-driven recommendation to implement maybe the precision pest management strategies that target peak activity periods and high-risk locations. Thank you so much.
Good afternoon, everyone. My name is Suman Prajali, and I'm working in Dr. Raymond's lab at the University of Alberta. Today, I'm going to talk about my research on phenotypic and histopathological evaluation of the canola lines carrying clubroot resistance from Brassica oleracea. Okay, this is the one, right? So, okay, let's get started. Why does clubroot resistance matter in canola? So the clubroot disease is major disease in canola. It causes significant yield reduction in canola. And most of the Canadian cultivars that are grown in Canada, their resistance against this disease is derived from Brassica rapa, which provide A-genome resistance. So the issue with this source of resistance is it is pathotype specific, and it can be easily broken down with the new virulent pathotype. So our research is basically seeking alternative source of resistance, basically from Brassica oleracea. And so our objective of our research is to test canola lines carrying a resistance source from Brassica oleracea. So a little bit on clubroot disease and the gall formation. So clubroot disease is caused by biotrophic pathogen, meaning it depends upon host tissues for completing its life cycle. And it is soil-borne disease. And also, it is monocyclic disease because it completes only one cycle per growing season. And there are basically two different infections, primary infection and secondary infection. So primary infection starts when those dormant resting spores, it germinate into biflagellate structure, and it get infect the root, primary root cell. So, and then secondary infection when those secondary zoospore infect the cortical tissues. So those stages are the secondary infections stages. And understanding these primary and secondary infections are important to understand the disease resistance of those introgressed loci from Brassica oleracea. So our plant materials consist of 3 canola lines carrying clubroot resistance from Brassica oleracea. And we inoculated these 3 different canola lines with 10 different pathotype of clubroot. And we did disease assessment at 45 days post-inoculation. And for histopathological assessment, we collected root samples at 28 days of post-inoculation. And we fixed and sectioned those root samples and also stained for microscopic analysis. And we compared the infected tissues against control. So our results was quite interesting, and it shows those canola lines carrying clubroot resistance loci from Brassica oleracea showed significantly lower disease severity. As you can see in the figure, the y-axis, it represents disease severity in percentage, and in x-axis we have 10 different pathotypes. So as you can see, those disease severity across among those 3 genotypes are almost close to 0. It indicates that those resistance loci from Brassica oleracea is basically providing broad spectrum resistance against all these pathotypes. So canola lines carrying, also we found that canola lines carrying clubroot resistance of Brassica oleracea showed lower cortical cell infection and no visible gall at 28 days post-inoculation against 3S pathotype. As you can see, the figure in the left, the y-axis represents a percentage of infected cortical cells, and the x-axis it represents the genotypes, 3 different canola genotypes. So we found like those cortical infections was basically reduced, and it ranges. It was like less than 30%. And the figure on the right on the top, it shows those tissues level, those cortical infections among those 3 different canola lines, and were compared against control. We found that those resistance lines were effectively inhibiting those clubroot infection. And also, our canola lines shows no gall at 28 days of post-inoculation. So overall, this indicates that those resistance loci that were introgressed into canola lines were inhibiting those cortical infections. So to summarize, the canola lines carrying clubroot resistance of Brassica oleracea, it shows lower disease severity index. So it inhibited the secondary infections, and there was no any visible galls. So it's basically providing us like durable and broad spectrum resistance against clubroot disease. So the practical application part of our work is these canola lines carrying clubroot resistance loci from Brassica oleracea. It can be used as a new source of resistance for breeding. So we'd like to acknowledge all these funding agencies, Agri-Food Canada, Radar Canola, Alberta Canola, SaskOilseeds, and Manitoba Canola. Thank you.
Yeah. Good afternoon, everyone. My name is Gursimran, and I am a master's student under Dr. Harmeet Chawla at the University of Manitoba. So I'm working on epigenetic changes in canola under drought and Verticillium longisporum. So to begin with, what if I tell you that plants can remember stress without bringing a change in their DNA sequence? That means that what if the plants, they change their DNA side change, and in that process, they remember a stress which they previously were exposed to. That's what epigenetics is. So epigenetics is any change on the top of the DNA, which means a change in the DNA side chain without a change in its nucleotide sequence. And it has the power to change the gene expression, which means that it can turn a gene on and off. And as a result, it can also change the phenotype of a plant. So why epigenetic studies are important? Because it's just like if you consider DNA as a manual, then epigenetic modifications are those tags on the DNA which help you to know that, okay, this page is important and it needs to be read more, whereas this page is not important, it can be skipped for now. So basically, it controls that which gene has to be expressed and when it has to be expressed. Then it also helps the organisms to respond to environmental stresses, making them more resilient. And it also allows for heritable changes. So my research is on epigenetic changes in canola under drought and Verticillium longisporum. So plants are not exposed to one stress in the field conditions. They are always exposed to more than one stress. I'm working on drought and Verticillium longisporum, and to our surprise, based on the preliminary results that I got from my colleague, we observed that based on the visual scoring, we observed that the plants that were exposed to both drought and Verticillium longisporum actually performed better. And this is the visual scoring graph, where we can easily see that the plants that were well watered and were exposed to Verticillium longisporum were not, they did not grow that better as compared to the plants that were under drought. These are the pictures that are taken. So on the left-hand side, we have the plants that are under drought and Verticillium longisporum, whereas on the right-hand side, we have the plants that are exposed to this drought. And we can clearly see that the disease severity is much lesser on the plants which are exposed to both drought and Verticillium longisporum. To add on, we have the epigenetic results. So what we found out that there were some genes which were which helped the plant to become more stress resilient, and they were expressed more under the dual stress condition, that is under drought and Verticillium longisporum. And there was one of the genes which actually helped the plant to thicken its cell wall, and ultimately the plant was more stress resilient. So there are so many other genes. For example, there are some genes which are involved in iron homeostasis and many more genes which make the plant more stress resilient, and there is less disease severity under drought conditions. So the take-home messages for the research is that the disease severity is lower under drought conditions as compared to the well-watered conditions. And the epigenetic changes are also, they add more information to this study. And at last, I would like to thank my funders, that is SaskOilseeds, Manitoba Canola Growers, WGRF, and the Government of Saskatchewan and NSUG. Thank you.
Good afternoon, everyone. My name is Virginia Miguel. I am a postdoctoral researcher at Queen's University. So I'm coming from Ontario, so I not get used to this winter. And today I try to be fast as possible. And maybe you're familiar with these diseases that are clubroot, blackleg, and stem rot. These can be the most devastating and terrifying forces on the field, and that can slash canola yield over different percentages depending on the provinces, as Dr. Ali Reza explained that this morning. And as researchers, we use the power of biotechnology and also genetics approaches to understand the plant world. And particularly in my research group, in Monahan Lab, we study plant immunity and how to improve immunity. So with these genetics approaches, we can target specific genes to help plants or crops to cope better or better cope with different biotic stresses. But the major challenge is which gene to work with, because there can be plenty of possibilities. So for that, today in our research is about a promising family that is calcium-dependent protein kinases, or CDPKs, and today I will talk more about CPK28. So CPK28 is a calcium sensor, and in Arabidopsis it has been shown or studied that plants have mutant version of this protein or don't have an active CPK28 are more resistant to diseases, particularly if you see the infection of Pseudomonas syringae, these plants are more resistant. And then if you measure the ROS reactive oxygen species or ROS species in the apoplast of the plant cell, you can see that these plants release more ROS. What that means, we use ROS as a quick or early readout of the plant immunity because but in general, when you elicitate the plants with different pathogens, you can see the plant release this species in a way of kill the pathogen. So when you have in general more ROS, you have more resistance in general terms. Opposite to the mutant plant, we have the overexpressor plants. And the overexpressor plants are more susceptible to Pseudomonas syringae infection and also release lower level of ROS. Importantly is that neither the CPK20 mutant or the overexpressor plant present developmental trade-off. If we are thinking about breeding, this is an important trait to consider when we are trying to improve immunity. So CDPK is a very huge family, and I don't have to mention that canola is allotetaploid, have a complex genome. So we studied this big family in these plants, and we found out that for one copy of the gene from Arabidopsis, we can have between 4 to 10 copies in Brassica napus. So for that, I studied a lot this family, so we compile this information in a review that you can have the QR, feel free to scan it, and you have more information about this family. But to our business of today, I will talk about the subgroup 4 that CPK28 belong. We found that in Brassica napus or in canola, we have 10 candidate genes that can be ortholog of this CPK. But only 4 of them are strongly expressed through all the plant. So we moved forward with those 4 to generate CRISPR knockout lines. It was a lot of journey about how to do CRISPR, so I'm happy to say that I am obtaining my first lines and in this spring, and maybe for the next conference I can talk about more of that. But today I will talk more about, because while I was doing the CRISPR knockouts, we generated the overexpressor plants of these CPKs, and in this case, we elicitate this plant with, in this case, L18, that is an elicitor of a bacteria, and we found that the overexpressor plants, as expected, have lower level of ROS, something that we were expecting because we are overexpressing this protein. Then in our early results, we also test this plant, we assist the clubroot infection, and we found out these overexpressor plants are more susceptible to the disease. So together these results are telling us that we are actually working with a very good candidate, or a strong candidate that can help us to generate very elite canola for the future. I only have time to thanks to my supervisor, Jacqueline Monahan, and also our collaborator, Eder Lopez and Marvel Monte, and a special thank you also to Marcus Samuel that helped us or teach us in his lab how to do canola transformation. Of course, to our funder, that thanks to them, I can be here and studying this project, this fascinating research. And I have a poster number 8, so if you have questions, please reach me there. Thank you very much.
I'm Zhi Hongdu from Dr. Mark Smith's lab. We know that the seeding date for canola is very important. Those seeding late in the spring can expose canola to summer heat stress and fall frost, which could lead to poor seed quality and low production yield. Early seeding, on the other hand, can improve yield to some extent by providing sufficient soil moisture for germination process and also avoiding heat stress in summer. But early seeding, however, can introduce some challenges. The cool soil temperature can slow the germination process and delay the seedling growth. And the fragile seedlings can later be damaged by flea beetles because of slow growth. So considering those limitations, our project is to investigate the physiochemical process that occur during co-germination and seedling growth in canola, aiming to find some solutions to tackle those issues. So, considering the complexity of the germination process, for feasibility, we want to be more focused instead of investigating a large, broad area. So we think that membrane lipids could be a good research target because previous studies have shown that membrane lipids are associated with how plants adapt to temperature changes. So specifically, the plants modulate the lipid composition to help themselves adapt to the temperature fluctuations. So that's how they adapt to it. So we think that in our project, membrane lipids could be a good research focus. So for our experiment, we use double haploid line of canola as our major seed material. And we also use Camelina as an experiment reference because Camelina shows better co-germination performance compared with canola. And we also want to find some similar or divergent patterns through comparing the experiment results from those two different species. So hopefully this will enable us to find some interesting stuff. So for our germination process, it was conducted at normal temperature and cold temperature. And also the whole germination process was conducted at dark condition to exclude the effect from photosynthesis process. So for the seedlings, we regard the seedling with similar root lengths at both temperatures as at the similar comparable developmental stage. So the tissues, including hypocaudal and the roots from those seedlings, were collected for lipid extraction and RNA extraction, using for the lipidomic analysis and RNA-seq analysis. So our aim is to, by integration analysis of those two data sets, we can find some genetic factors within the lipid biosynthesis pathway or beyond the pathway. So and then we can functionally verify the roles those genetic factors played for co-germination process in transgenic canola. So currently, our poster shows partial of our initial results. So by analyzing the lipid composition and data, and also by analyzing the expression level of the genes involved in the lipid biosynthesis pathway, we finally found two genetic factors, which is ADS2 and FAT3, that could be contribute to the temperature-dependent membrane lipid composition changes. So the next step for me is to clone those gene copies out of these two genetic factors and then test them in the transgenic canola limes. So if you are interested in more details about our project, please come and stop by poster 3. Thank you.
Thank you very much to all our Flash Talk presenters. Let's give them just another round of applause for all of them. Okay, and for everyone here in person with us in the audience, they do have the posters just over here. They'll be available both today and tomorrow, where we have the other even-numbered posters being presented in front of them, and then potentially also on Thursday as well. So check that out if you want to learn more, as well as hear from the other posters that we didn't have presentations today. So now we're going to step into some presentations from our provincial canola associations. And so I might as well just introduce all three of them at the same time. So we'll start with Jared Bento from the Manitoba Canola Growers Association, followed up by Autumn Barnes from Alberta Canola, and then lastly, Doug Heath with SaskOilseeds.
Hi everyone, Jared Bento. I'm presenting on behalf of Manitoba Canola Growers and also on behalf of our research committee consisting completely of farmers, Manitoban canola farmers. These canola farmers represent the priorities and especially the critical goals of all farmers, all Manitoban canola farmers, with a specific focus on research. Many of those goals revolve around canola yield potential and stability through genetic and agronomic solutions, protecting yield from current and emerging pests, nutrient use efficiency through 4R, management practices, improving environmental impact of canola production, and equipment optimization, including reducing harvest and storage losses. Specifically, in 2025 and 2026, with our updated research priorities, we're focusing on things like verticillium stripe and blackleg, reaching the top of our priorities as the rise of disease has risen in Manitoba. Yield stability in environmental extremes, opportunities and risks in canola market profitability, integrated weed management, nutrient use efficiency, and flea beetle management. We're gonna be going through like a flash talk of each of our main areas in Manitoba Canola Growers research, focusing first on the research product funding and the applied research programs. These constitute 86% of our research funding categories. As I said, diseases constitute a major part of that focus at MCGA. Up to 42% of our funding goes towards research on diseases, followed by 22% on insects, 13% on genetic improvement, and less than 10% for the rest of those categories. Focusing specifically on the actual project funding in different funding initiatives, through CARP, we've, in 2024-25, we focused on, or we funded 5 new projects, 2 of them on verticillium stripe, 1 of them on sclerotinia, 1 on flea beetles, and 1 on heat stress. Another project that we started out on is actually something that you heard just earlier from University of Manitoba. This is the Applied Verticillium Research Initiative through Dr. Harmeet Chawla and Mustafa. This runs alongside MCGA's on-farm research program in collaboration with Dr. Chawla's lab. We focus on these on-farm trial types to use quantitative data on the amount of verticillium present in the soil, allowing us to correlate verticillium spore load to disease severity and yield, which you saw in, very clearly in Mustafa's graphs. This allows us to also correlate harvest management practices to the next season's spore load, which is the main focus of those projects and what we look forward to seeing in the 2026 2026 season. We can also compare this to historical data management practices from farmers, including crop rotation, diversity, fungicide use, chaff management, and harvest management. I'll switch now to that on-farm research program I mentioned. So this has been a major focus of our applied research and a major focus of my time here with MCGA. So this started in 2022 with 12 trial locations and these 3 trial types that were put forward by our Farmer Research Committee: nitrogen rate, seeding rate, and bioinoculant. Now, the goal of this on-farm program is to collaborate with farmers, agronomists, and researchers in providing the most relevant data and information for the productivity of the canola production in Manitoba. From 2022 to 2025, we took in the opinion and the input of our farmer board and other farmers that we've spoken to in our extension events to develop this program and add many more trial types, including now this year our 2 trial types that are focused specifically on early season fungicide and harvest management. Overall, since 2022, I'm glad to say we've worked towards 122 individual on-farm sites over 4 years. I'd like to share just one, there's that map of those 122 sites over 4 years, and also a special thank you to our on-farm research program partners, the research agronomists that we've interacted with all through the program. Just want to share one outcome from this year's seeding rate trials. Nothing out of the ordinary. There were 2 sites with increased and decreased yields and 2 with no significantly different yields. But the highlight I want to make today is regarding our new focus, or our focus specifically on farmer profitability in the on-farm program. Not only in research, we'd like to make this focus, but now going forward across all communication paths, we'd like to focus on this farmer profitability and making it very clear and direct. So for all on-farm trials going forward, we're including this economic analysis table that you see here, which estimate the farmer's profit across all treatments using summarized data from Manitoba's cost of production guide to compare yields of input to input costs. Our growers always look to the bottom line in all of our communications, and this is part of how we aim to make sense of theirs. Also glad to say that the Manitoba Canola variety trials have been published for this year, for 2025. We've completed the 2nd year of this canola variety trials program, tested 15 canola hybrids from 5 seed companies at 8 testing locations. And that information is now publicly available to anyone who would be interested. We've also updated Manitoba's Canola Lab Diagnostics program. We are continuing with free testing to help Manitobans growers identify and manage crop diseases. The MCGA's Crop Diagnostic Services provides molecular analysis and interpretation for actionable results to support farmers in their in-field decision-making and also boosting productivity. The tests include blackleg race ID, verticillium stripe content in tissue material, and club root. We have some postcards floating around, actually over by the coffee station there. If you'd like to scan this QR code, or you can just search Manitoba Canola Diagnostic Services to learn more about the sampling protocol and the results reporting. And finally, going beyond just actual research projects and services, a little bit more to extension. This year marked the 4th annual Canola Research Camp, where our goal is to bring together a group of researchers to improve the capacity of the research community and provide quality interdisciplinary research projects and results for Manitoba canola farmers. So this year, we brought together 10 researchers from 5 institutions and 6 disciplines to visit 6 unique farms all across Manitoba. I just want to share 2, I think, impactful quotes from some of our attendees saying that being on the farms and seeing things in action is a great way to touch base with the people for whom we do our research. And I think that's just something that got me into research in general, is research in agriculture in general, and something that I try to remind myself is that the end goal of every dollar, every hour of research is eventually meant to be for the farmers that we interact with. Another attendee said they came up with a bunch of ideas that they want to tackle and try out. An unexpected one we saw was that 2 researchers, both working on verticillium, in the same plant science building, didn't know each other were, and now these researchers are going to use their insights for the priorities going forward. So that takes it for me, and I'll pass it now off to my next provincial research representative.
Hello again. I'm just going to wait for my slides to come up, but I introduced myself earlier. My name is Autumn Barnes. I'm an agronomist and a contractor covering Brittany Vischer, who's the Director of Research for Alberta Canola. She's on maternity leave, so I'm covering that contract for the year. If you do have questions about canola research in Alberta, please feel free to get in touch with me before February, and then Brittany's coming back in February, and I'll move on to another contract. So I'm going to give a bit of an overview of our funding process and some of the research activities that I've been working on this year. So first off, this is the Alberta Canola Research Committee. Alan Hampton is in the room somewhere. Saw him a little while ago. There he is. Great. So make sure you tackle Alan, ask him with for questions later. Cheryl Westman and Justin Aninga are from Central Alberta, and then Andre Harp is the chair of Alberta Canola, and he lives and farms in the Peace region. And I should mention as well, Alan Hampton is farms in near Drumheller and is also on the WGRF board. So the research committee at Alberta Canola exists to find the most sustainable and efficient ways to grow canola in Alberta, which includes identifying best practices for pest management and other production problems. This also includes supporting with research, researchers engaging with them, identifying research gaps, which is something that we're trying to kind of lean into a little bit more. And then when we find those research gaps, try to fund research in that area. And these objectives were developed by the Alberta Canola Directors, Board of Directors, and they fit within the greater strategic plan that Alberta Canola is working on right now. So the three main activities that Brittany and myself work on in this position focus on three core areas. So research funding and administration takes up the majority of my time. Research engagement, so this is anything that kind of brings researchers out to the farm or involving them in farming priorities, involving them, getting them together with farmers, kind of sort of like what Jared was talking about, but we're doing stuff a little different in Alberta. And then also participating in a number of different committees, steering committees and things like that. Canola Council, I guess we'll be talking about those in the next few days. So hopefully we're good here. At any point in a year, Alberta Canola has about 30 active projects that we're funding. So with active projects, we have to review interim reports, we have to approve them, ask any questions, review financial statements, approve payments, and then eventually when the project is done, review those final reports and close it off, put it on the website. So each year we'll have new projects start and we'll have projects that finish and come out of our program. So when projects come into the Alberta Canola Research Program, they start as letters of intent. This year, I reviewed about 100 LOIs, and then from that, chose which ones would be the highest priority for farmers in Alberta, which ones will have the most impact, and cut those down to about 28, 29 this year. So our full proposals get reviewed by the Research Committee, which I showed earlier, as well as a number of external reviewers. So those are folks who have some sort of technical expertise in whatever area the project is in. We have a review process, co-funding groups also review, and then we get together and talk about which projects we think will have the most impact for the limited budget that we have. So based on that, those full proposals are then ranked and voted on by our research committee, and then there's a bit of negotiation that goes back and forth between co-funders, and we decide which projects are actually going to get funded, and that's kind of happening right about now. So I mentioned that I started with about 100 LOIs in the spring. Of those, we look at kind of 3 core funding areas. The Agriculture Funding Consortium, which is based in Alberta, it has about 20 different funders that get together and fund different types of projects. The Canola, and it's administered by ARDAR. The Canola Agronomic Research Program, or CARP, is administered by the Canola Council of Canada. And then we also contribute sometimes to projects under ADF, the Agricultural Development Fund. We also fund projects through the Agri-Science Research Cluster, the federal five-year funding call, but I don't have that on here because it's not a regular annual project or call. So, of the projects we're looking at this year, our potential co-funders with Alberta Canola would be SaskOilseeds, Manitoba Canola Growers, ARDAR, Alberta Innovates, WGRF, and ADF as well. And when we co-fund, we can stretch our dollars even further, so we're really big on leveraging and trying to get the most out of every Alberta Canola research dollar because research funds are limited. And then, yeah, so we're in the middle of figuring out how many projects we're going to fund. Usually it works out to 10 to 12 new projects a year out of those 29 that we're looking at right now. I'm going to talk a little bit about research engagement now, which is something I get a lot of joy from. So this year we did a research tour in southern Alberta. And so this was really to link together research funders, farmers, and industry and try to get us all on the same page about the different constraints everybody has, try and understand regional challenges, different opportunities, what kind of capacity exists. And then also build those relationships. So I live in Lethbridge. My career has been spent in southern Alberta, so it's pretty fun this year to be able to bring a group of around 30 funders, researchers, and industry folks to Lethbridge for a couple days and look through some sites. So we went to research sites at Ag Canada Lethbridge, Lethbridge College, Farming Smarter, and the University of Lethbridge. And then we also went out to farms and industry sites. So this is actually at an irrigation farm in southern Alberta called Quattro Farms. And they, in addition to seed canola, also have a number of different specialty crops like catnip and peppermint. They also grow potatoes and sugar beets and things like that. And so this is right outside of their still where they actually bring in the peppermint and the catnip and they distill it and they sell it as essential oils. This is a good money maker for them. We also stopped at Kings Lake Colony and Cronkite Ag at Foremost. So Foremost in southeast Alberta, or southeast of Lethbridge, I guess, is a really, really dry area. And southern Alberta has been really historically dry, but also we've kind of been in a pretty massive drought cycle. So getting funders together to come down and actually put their feet on the ground and understand some of the constraints that dryland farmers have was really impactful. You know, how farmers make decisions on if they're going to include canola in their rotation, because for many years in the past it just hasn't been economical when you're looking at the types of yield potential we have when you're only getting a couple inches of rain in a year. So that was really impactful. We stopped at the Bayer seed production site and talked to one of their breeders about the process of even getting these traits that Alberta Canola research dollars fund the development idea of these traits and then getting those traits into the commercial varieties that a farmer is then going to buy. We had folks from the St. Mary River Irrigation District and the Alberta Irrigation Districts Association as well, talking about how irrigation even works, which is pretty cool. There's a lot to it, the history of it. And then my favorite site, because my first real farm job was on a dairy farm, so we stopped at a dairy farm that milks about 350 purebred Holsteins and learned about how they use canola meal in their feed, in their rations, and then the general operations of big, modern, well-run dairy farms. So it was really cool and kind of like a 360 view of canola and the wider agriculture industry, and a really good opportunity for us to work together. Another thing that we're working on right now, so one of the things when I got brought on this contract this year, with a new strategic plan with Alberta Canola, they wanted to get some momentum behind a research plan. And so I started asking, okay, well, when have we gone to farmers and really asked about the type of projects we want to fund? We've asked about research priorities, which is really good, but also, you know, should we be investing more in agronomy versus genetics, applied research, niche market opportunities, or bigger market development type opportunities? How should our research results be shared? You know, do our levy payers have strong feelings about research? Do they want us to just take it and run with it? So I worked with internal Alberta Canola staff to build this research survey that is happening right now in Alberta for farmers. So probably not a lot of you in the room that might fit, but we're going to have results for that in the new year, and are looking forward to sharing that and then using that to kind of build a more fulsome research program going forward. I don't have it on the slide, but this survey, like a big survey like this, is a big undertaking, and it was supported by ARDAR, so we're really grateful for their funding to help actually get out, and we've got a lot of really great responses, and I'm excited about it. This is my contact information. I like to brag, I just ran my first marathon this year, so this was right after I ran 42 kilometers in the rain, so I'm going to give myself a pat on the back. My email address is there. You can reach me until the end of January at that address, and then Brittany Vischer will be back in February, so feel free to send her an email and welcome her back when she does come back. And thank you for your time. I'll be here all week, so happy to take questions later.
All right, good afternoon. I'm Doug Heath, the research manager with SaskOilseeds, and I'll be giving a quick overview of some of the research and programs going on at SaskOilseeds. So the first is our disease testing program, which we offer to registered growers in Saskatchewan. So we had a slightly higher uptake of blackleg and verticillium testing this year compared to 2024. And we saw a reduction in the percentage of samples with blackleg compared to the really bad blackleg year in 2024. But we also saw a large increase in the number of verticillium positive samples compared to last year. So out of 164 samples that were submitted, 41 were positive for L. maculans only, 34 for verticillium only, 46 for both pathogens, and 43 were negative for both. And similar to what Alareza said this morning, out of 18 soil samples, we didn't find any clubroot positive samples this year. So as part of our agronomy-based research, we also hosted canola disease ID training days at 4 of the agri-ARM sites in Saskatchewan. So this is the second year that we offered this program. So it's a good way for agronomists to get hands-on disease sample collection and diagnostic training. And that focused on the 4 major canola diseases. Okay, so that was the first of 8 demonstration projects at agri-ARM sites in 2025. So the final data analysis and reports will be ready early in the new year. And these included, do micronutrients applied to canola actually increase yield consistently? We'll talk a bit more about sclerotinia insensitivity to fungicide in canola. So this is independent from a couple of other projects on that topic. Does seeding date and rate of canola affect spring flea beetle pressure as well as seed yield and quality? So that's the 3rd year that we've done that project. Looking at potash for replacements for chloride uptake. Looking at response of enhanced efficiency nitrogen fertilizer products and blends versus urea for canola establishment, yield, and quality. That's the 2nd year we did that one. And for field scale research, we looked at the economic comparison of spot spraying versus blanket spraying of herbicides, as well as the impact of low versus high stubble heights of canola and wheat, which was co-funded by Sask Wheat. All right, and our on-farm research trials program is called Top Notch Farming, and it expanded to 29 sites with 6 protocols in 2025. So we continue to work with Kayla Slind at Work, which is one of the agri-ARM sites. And she manages the protocols, the communications between contracted agronomists, and ensures all the correct data is collected and does the data analysis. And several of the different Sask crop commissions work together. They have their own on-farm trial programs, but we work together in this sort of combined program. So this year, those groups, including SaskOilseeds, worked with Kayla and Rob Workington with Workington Ag Services in Davidson, where we hosted a joint field day. And so that included one stop for each of the different crop types. So the participating cooperators and agronomists will get first access to all of that on-farm trial data at a banquet in February. And then the data will be released publicly after that. So we do have results from the 2024, which had program which had 4 protocols, but due to the environmental conditions that year, there weren't a lot of statistically significant results for any of the trials. The first one is split or top-up nitrogen trial. So there's 2 options for the growers to choose. So there were significant trends observed between fertilizer treatments and protein level, as well as fertilizer treatments and moisture content. But it was only a trend for the 70% N at seeding plus 30% in-crop top-up, which provided the highest yield and greatest net return. For the enhanced efficiency nitrogen fertilizer trials, 3 different sites used different fertilizer products, and one of the sites used different treatments. So we are able to work with each cooperator to customize the protocol so that it works on their farm. And yield differences were observed between the sites that used different treatments. So it's suspected that there's differences due to location. For the seeding rate trial, locations were grouped by plant count densities, and there was variability seen in the results. So a couple of the sites saw the higher yields with higher seeding rates, and one site showed the opposite effect. For the foliar applied nitrogen fixing biologicals, it was a dry year, and on 8 locations where 2 of those sites used Envita, 5 sites used Nutricia, and 1 site used both products. But due to the conditions, there's no statistically significant differences in plant densities, yield, or grain quality for that year. So this is just a summary of the locations and the 6 protocols that were done in 2025. So 3 of the protocols were repeated from 2024. So that's the split or top-up nitrogen, the enhanced efficiency nitrogen products and seeding rates. And 3 new protocols were included for sclerotinia fungicide applications, boron applications, and blackleg fungicide. And again, the results will be ready in February. So looking forward to next crop season, we hope to increase by a few sites. And we're going to add a couple of other protocols as well, including verticillium stripe management, so similar to a Manitoba Canola Growers program to compare swathing and straight cut effects on verticillium. And SaskOilseeds amalgamated with Sask flax, so we are doing some flax for fertility as well. And depending on the amount of hail, we may open up a hail recovery methods protocol. And so we hope to expand further into the east and southern parts of the province. So if you're a Saskatchewan producer and interested in on-farm research, please reach out about becoming a cooperator. All right, so we consistently have about approximately 100 active projects each year, and those fall under the 3 main themes of agronomy, germplasm development and trait discovery, and canola utilization. So we co-fund those through competitions like Saskatchewan's ADF, the CARP program, Alberta Funding Consortium, Canola Agri-Science Cluster, NSERC, and MyTax. So there isn't really time to summarize 100 projects, so I'm just going to briefly go over just a few last slides here. So we are co-funding several projects with at least some aspect of co-infection with blackleg and verticillium. And so more work is needed to determine whether some or all of the R genes for blackleg are being broken down by the co-infection of verticillium, or whether certain environmental conditions are making blackleg worse under co-infection. So that could make it more important to test for which blackleg races are in farmers' fields. We're also looking at a couple of different projects, one through the canola cluster and one through CARB, looking at a total of six fungicides and the baseline for insect sensitivity by sclerotinia across the prairies. We're also looking at susceptibility genes, so S genes for proteins and receptors that pathogens recognize and require for infection to progress in the plant. So by knocking out these S genes by gene editing or screening mutant populations, we can discover lines that are resistant to the major diseases. So we saw great success with that with Wei Xiao's project with clubroot S genes. And now we're looking at strategies in current projects and possibly new proposals for clubroot, verticillium, and blackleg. And I'm out of time, so I'm just going to hurry through these last couple slides. So we're looking at bringing in genetic diversity from diploid lines for verticillium clubroot resistance, as well as heat and drought resistance. And this is the last slide. Also looking at herbicide-resistant weeds on multiple projects. So please check out SaskOilseeds.com and the Canola Council's Canola Research Hub to learn more about the research projects that we're funding. All right, thank you.
Well, thank you very much to our research managers to give us a nice prairie-wide perspective on what our grower groups are providing value for their members. And again, thank you. And in lieu of gifts, we are making a contribution to the Friendship Inn just down the road here. And thank you for taking your time on your busy schedules to have these presentations as well. So I'm going to hand it over to Jay now to kind of wrap things up here and take it away.
Mic one? Okay, we're working. All right, so last year we tried this. We're going to do it again. We're not even going to sit down. It's going to be so fast. But 3 farmers are going to come up, one from each prairie province. Alan, John, you're coming up for me, and Jason. And I'm just going to check in. Come on up, Alan. Check in with all 3 of them about some takeaway from the day. So we're going to close with a quick farmer takeaway. And just while I'm waiting for these 3 guys, I just have 3 quick announcements that I was going to say afterwards. So right after this, 4:30, we have a reception for, I think you needed to register for that. So if you did register, you can head over to that. BASF is the sponsor. And Clint, you're key. Clint, I think you're making a short presentation over there. Just say yes. Okay, and then dine around. There might be a couple spots left, but thank you everyone for signing up. We had really good turnout. The reservations are under my name, Jay Wetter. So if you could go as a group, you could meet. I think the reservations are for 6:30. Meeting might be problematic, but you know where your restaurant is. So just go there for your reservation time and say, I'm here for the Jay Wetter group, and they'll seat you. And then, so tomorrow morning, I'm going to save this till we're done. Alan, how you doing? Alan Hampton from Alberta. Fantastic. Okay, so what was one thing you I said one. That's a lot. You can read your whole napkin. You can read your whole napkin. So what did you pick up from today? Here.
Well, I think the bottom line, Curtis nailed it right out of the gate. He talked about innovation, creating value, and critical for the success of the industry. Well, as I listened to every presentation on disease, herbicide resistance, climate challenges, trade difficulties, you name it, we need to continue to support research and innovation in order to keep our industry strong and competitive. That's probably a fairly generic sort of response, but as a farmer, we need this type of work going on, and as a farmer, I need to support it because it's the only way that we're going to probably overcome the challenges that all of us are facing. So that was my takeaway for today.
So a broad takeaway that we need to fund research. Okay, big picture guy. John, did you go big picture or did you go specific?
We started the day talking about geopolitical issues and demand issues, which definitely affect farm economics. And so that's kind of, it's with that lens that I kind of come to this conference, you know, how is research going to support the economics on my farm and make me sustainable and survivable for the next generation to take over. My takeaway today came out of the weed panel. I thought that was excellent. Really good presenters, and it was Andre that had a comment. He made a comment here about chemical/technological transitions exposing some of the gaps, and that's going to be something I can take home and maybe talk with some of my peers about. We have this, the economics of farming is driving the scale of the farm. We're getting larger, covering more acres, but in that, how do we still do patchwork farming where we're looking after the little things which can become big issues? And then I liked your comment, Jay, like, is there research that we can do that can focus on changing farmers' habits? Because sometimes we get so caught up in the efficiency of the farm that we forget about, maybe best practice. And, and I have had my agronomist tell me for the last 10 years, more water, slow down. It's something that we've adopted, but it's not widely adopted because I don't think there's great research that supports that. So.
Right on. Thanks, John. So Alan Hampton farms in Alberta, director with Alberta Canola, the research chair. Autumn had your picture up there. John Fair farms in Saskatchewan. What's your hometown, John?
West Central, so Herschel, Rosetown, Kindersley area, West Central.
Thanks, John. And Jason Kaler, who's the rep for Manitoba Canola Growers. Jason, you're just north of Carman. Elm Creek or Carman? What's your hometown?
Halfway in between.
Okay, what did you pick up from today?
Reiterate what these guys are talking about. I enjoyed listening to Chris talk about canola's role in renewable fuels this morning. I think that's also a very important part of our industry. I think there's a big market emerging there. And again, the weed panel, definitely some meaningful conversation there. I feel like, again, more water, slow down. You talked about making guys twitch at 10 gallons. We spray all our glufosinate at 15 at 10 miles an hour. So that's what we try to do to make ourselves efficient. We think it helps. So, and all the weed resistance and everything. Yeah, great day.
Thanks to all three of you guys. Good to see you, and we'll visit again later. So the last quick announcement is, so tomorrow morning, continental breakfast at 8, opening remarks at 9, and then just a reminder that there will be a presentation and discussion about the canola steering groups from 8 till 9, so in that breakfast window right here. Over in that corner? Okay. So if you want to talk about the steering groups tomorrow at 8, over there. All right, well, thanks, everybody. I'll see you over at the reception. And then, like I said, if you signed up for Dine Around, it's under my name. Jay. Okay, bye-bye.